Hello and welcome back to part 3 of Histology of Nerves, Vessels and Lymphatics. This is actually going to be a little bit of revision for you, hopefully welcome, because I find that the nervous system is an area that often occurs around the mid-semester exam, and so students are more focused on the mid-semester exam than they are about learning about neurons and nerves and so on. So this will be a little revision of nerves. Of course, when you do dissection, you'll be encountering nerves, and this is how to identify them histologically in section, what they're made up of, and what the different parts are present within the nerves. So the objectives for this part 3 is that you should be able to identify and describe the normal histological structure and function of peripheral nerves, including as part of a neurovascular bundle, which we just talked about in part 2. No, part 1, wasn't it? Yeah. Anyway, you get the idea. So you may remember, or you may not, depending on how much effort you put into the nervous system as a tissue, nervous tissue lectures, that a peripheral nerve consists of axons, which may either be myelinated or non-myelinated or unmyelinated, doesn't really matter which term you use, that the cells that perform the myelination, or actually the cells which are associated with the axons, whether they're myelinated or non-myelinated, are of course the Schwann cells, one of the two types of glial cells which exist in the peripheral nervous system, and that there's connective tissue layers on the outside, and that's secreted by fibroblasts. You'll recall that there are connective tissue layers around peripheral nerves. The outermost dense irregular connective tissue layer around the nerve is known as the epineurium. So that contains fibrocytes on the outside of this dense irregular connective tissue. Then you get bundles of axons in a structure known as a fascicle. And around the outside of that fascicle is another layer of dense irregular connective tissue. This is known as the perineurium. So epi means upon, peri means around. So around the fascicle we have dense irregular connective tissue called the perineurium. And then around the Schwann cell and its axons associated with it is a layer of loose connective tissue, which is known as the endoneurium. So the loose connective tissue is never directly around the axon, it's always around the Schwann cell and its axons. It's either maybe myelinated axon or unmyelinated axons. And so that's the loose connective tissue layer known as the endoneurium. Endo means within. So here we've got several nerves in section. This is a trichrome stain section. So here blue is collagen. Out here, and unfortunately the blue is gone to sort of a reddy color. So this outer layer here is the epineurium, so dense irregular connective tissue. It sends in these folds of this perineurium, so surrounding a fascicle. So here's one fascicle, here's probably another fascicle, so this will be dense irregular connective tissue. You can see some blood vessels present here. And then around each individual Schwann cell, so here we've got a Schwann cell, here's its nucleus, here's its cytoplasm, here's our axon, and here's our myelin sheath. Remembering, myelin sheath is basically just a wrapping of the cell membrane, and being largely lipid, gets washed away for microscopy, so you often end up with this sort of pale region here. And this little blue line here on the outside represents the endoneurium, so the layer of loose connective tissue around the outside of Schwann cell and the myelin axon that it's associated with. So if we zoom in here, we can see here we've got the dense irregular connective tissue of the perineurium forming this fascicle, and here we've got all of these large myelinated axons here. So here's the nucleus of the Schwann cell, here's its cytoplasm, here's its axon and the myelin sheath. You often get this sort of funny little stripy like bike spokes around the outside. These are the interclefts associated with these myelin sheaths. And then out here we've got some nuclei of Schwann cells, and then these are all the unmyelinated axons. So they don't have those big thick bands around the outside. So these are the unmyelinated axons. So here's that cell that we're looking at there. Here's the nucleus of the Schwann cell, the axon, the myelinated axon, and here's the nucleus of a Schwann cell and several of the non-myelinated axons that it's associated with. Now, some of these nuclei may also belong to fibroblasts. Probably these darker ones, the smaller ones, are the fibroblast ones on the outside there. So for this example, this one here, you can see the collagen on the outside here represented in blue. So that's that one there. So here we've got our Schwann cell wrapping its cell membrane around the outside of the axon lots and lots of times, forming the myelin sheath. Under high magnification, we can actually see, this is showing an example here of the myelin sheath forming around the axon. You end up with this sort of record, LP record. I don't know if you guys know what that is. It's sort of made a comeback recently, the old LP records. So you may have seen one, but most of you are probably too young to know what an LP record looks like. They have grooves, and there's a little stick that picks up the bumps in the grooves, which then produces the music. Anyway, before most of your time. But it has this wrapping around the outside, so all of these white spaces are actually the cytoplasm of the Schwann cell, and the darker lines are the cell membranes around the outside. So here we've got our Schwann cell, just like the one we were looking at before. So here's the nucleus. So here's the nucleus, here's its cytoplasm, here's our axon in the middle, and then here's our myelin sheath, which of course under electron microscopy, lipids are often stained varying levels of electron density. Here it's quite, myelin sheaths are often stained very electron dense, so you have these black lines which under this higher magnification look like this. Remembering we wash away most of the lipids for light microscopy, so it has this weird sort of spoke-like structure. Now, also remembering that Schwann cells don't only associate with myelinated axons, but any unmyelinated axons are also associated with Schwann cells. They're just not myelinated by them, so they sort of nestled inside. For example, this axon is sort of nestled inside the cytoplasm there, just sort of resting in there like Nana giving a grandkid a bit of a hug. And so all of these unmyelinated axons are being held by the Schwann cells, just not myelinated. So out here we've got the collagen fibrils of the endomysium, and they actually have a little basal lamina on the outside of the Schwann cells as well. And then that binds with the loose connective tissue, holding it all together. So when we say endoneurium surrounds each individual axon, what we really mean is that the endoneurium loose connective surrounds each axon and its Schwann cell. So what do peripheral nerves look like in section? So what I look for is waviness. Peripheral nerves are often really wavy in appearance, and so that's what you're looking for. So here, and these are actually peripheral nerves. So there's a couple of things to look for. Look for this gap between surrounding tissue. This is often obviously artifact, but usually it means there's a dense connective tissue layer on the outside, a perineurium. So we've got a fascicle of axons here. You can see some of the axons in cross-section. Here they're longitudinally or obliquely sectioned, but when you do get the longitudinal sections, they often have this waviness associated with them. You can hopefully see some of that waviness over here as well. Here's that gap, then a layer of dense irregular connective tissue, perineurium. Here's that gap again. So that's what you look for to help identify peripheral nerves in section. Alright, so that's it. Just a quick overview and revision of the structure of peripheral nerves. So that ends part 3. Please take a bit of time now and attempt part 3 quiz to review your understanding of the content. And of course, please feel free to attempt these quizzes as many times as you like for your own revision purposes. And lastly, I'll see you in part 4, where I'll just pick some random slides and see if we can identify blood vessel nerves and lymphatics. See you then.